Are going to be downhill for the breakdown of the fatty acids. You can't reverse that energetically if you're going to synthesize the fatty acids. So you can't do that simply from a thermodynamic viewpoint. The other thing that's different is that the synthesis of the fatty acids actually takes place in a different region of the cell. The synthesis occurs out in the cytoplasm, whereas we just saw that the breakdown of the fatty acids occurs in the mitochondria. So these two processes are carried out in different compartments of the cell. We will find that they use a different carrier protein. The breakdown uses the coenzyme A. We will find that the synthesis involves another protein called the acyl carrier protein. The other thing we'll find is that the entire process of the synthesis occurs on a huge enzyme complex that's kind of set up like a factory with the different steps in the synthesis carried out by different enzymes in an assembly line type of fashion. It's really a fascinating type of enzyme, and we'll have a picture of it a little bit later here. Now, the first stage you have to do is to get all of the building blocks out into the cytoplasm. And the building blocks, as we'll see, are going to be acetyl coenzyme A. That's the source of the carbons. We build up the fatty acids from the 2-carbon precursor acetyl, and so the fatty acids are generally even numbered of carbons. We also are going to need reducing power, and in the cytoplasm, the reducing power is NADPH. To get these raw ingredients out into the cytoplasm, we have a little cycle here in which the citrate from the citric acid cycle in the mitochondria is transported out to the cytoplasm. It then does some reactions that look kind of like the reverse of part of the citric acid cycle, but they're separate enzymes. And we form acetyl-CoA, oxaloacetate. That's reduced to malate. That is reoxidized to pyruvate and generating NADPH. The pyruvate goes back inside and proceeds with part of the citric acid cycle. The net reaction is we get acetyl-CoA on the outside and we get NADPH on the outside. Okay, now the energy problem is the biggest problem that needs to be solved here. We need to activate the acetyl coenzyme A molecule into a much higher energy molecule than we saw during the breakdown of the fatty acids. And this is done in the following way. We form something that's called malonyl coenzyme A. And what we do here is we start out with the acetyl coenzyme A. So here is our acetyl coenzyme A molecule. And we react that with the ATP molecule and a bicarbonate molecule, just CH3 minus 1, just a CO2 that's reacted with water here. And we generate then the malonyl coenzyme A molecule. And that has this structure here. So this is a molecule of the malonyl coenzyme A. And this is a high energy molecule, and we release ADP plus phosphate plus protons. And so each molecule of the malonyl uses one molecule of ATP for its formation. And that is the primary source of energy for the synthesis of the fatty acids. We also need reducing power, as we'll see in just a second. Okay? So that's what is occurring here to produce the energy. but this is not like kind of a priming reaction, because each molecule of the two carbons here that are incorporated into the fatty acid have to come from the malonyl coenzyme A molecule. So each molecule that comes in has to be in this form here. So now what I'm going to do is go ahead and go through the complete cycle here. And I'll be writing it out on the board here, but it's also given. Here is the activation that we just talked about. Here are the enzymes involved. And let me just talk about this before I go on here. This is the molecule that's called the acyl carrier protein. We are going to transfer this molecule of acetyl or of malonyl coenzyme A to the acyl carrier protein, which is part of this huge enzyme complex. This is a picture of the acyl carrier protein. It has this long phosphopanethine group on it with a sulfhydryl group at the end, and it simply transfers the malonyl group from the coenzyme A sulfhydryl to the sulfhydryl of the acyl carrier protein. And that's where we begin the actual synthesis step for amino acid biosynthesis. This is the whole process. I'm going to write it out on the board now in some more detail. And so you can see the detailed reactions that are occurring here. So we start by putting a molecule, one molecule of acetyl group on an enzyme called the KS. And I'll give you these abbreviations a little bit later. Put a little block around that. That enzyme has an active site sulfhydryl, and we transfer one molecule of acetyl to that group initially. So we have this molecule here. Acetyl attached in a thioester linkage to what we call the KS enzyme. Then we react that, we put on another group, the acyl carrier protein. That sulfhydryl reacts with our molecule of malonyl. So here is our malonyl group that we've just made. And what happens in this reaction here is the electrons in this bond attack that carbonyl carbon and form a bond between that carbon and that carbonyl carbon. The electrons on this bond go into that sulfhydryl group to release the free sulfhydryl, and the result of that reaction then is going to be the release of a molecule of carbon dioxide, and we are then going to have the KS with a free sulfhydryl group, and we're going to have the acyl carrier protein now with this molecule attached. So this is CH2, C double bond O, CH3. This is the critical reaction in fatty acid biosynthesis. This is the one where we put together the growing fatty acid chain. We started out here with 2 carbons on the acetyl group, 3 carbons on the activated malonyl group. We are using the energy of that high energy bond to form a new bond right there. And now this is broken. This piece right here comes down here. Let me just put some labels here. We'll call this A, B, let's call this C, D, and E. This now, the CO2 leaves as E. This is the C molecule, this is the D molecule, this is the A molecule, that's the B molecule, okay? Just to show you how those occurred. Now, that's the key reaction. That is the condensing reaction, where we put these 2 molecules together. We actually call that the condensing enzyme that carries out, that catalyzes that reaction. Now, the next step and all of the succeeding steps are going to look very much like what we just encountered in the breakdown of the fatty acids. Notice that we've got a ketone group here, so we're going to go ahead and reduce that ketone using a molecule of NADPH here. And so this is going to be NADPH, generating NADP+. And the enzyme that carries out this reaction, I'll just abbreviate it as KR. And that is going to generate the acyl carrier protein, which now has this group here. So we've simply reduced the ketone to an alcohol here, using as reducing power NADPH. I think you can see what the next step is going to go. We are going to eliminate water here. So there goes the water. And we are going to form a double bond here with the acyl carrier protein. And this is going to be CH, double bond, CH, CH3. We're going to carry out our second step of reduction here. So we're going to bring in another NADPH molecule, generate another NADP+ molecule, and this is now going to generate the fully reduced fatty acid. With the carbons here. And now what we are going to do next, so this is all the chemistry. And let's go through this again just to emphasize it here. This is the key step. We condensed this molecule with that molecule to make the 4 carbons, releasing a molecule of CO2. Reduced the ketone to an alcohol. Removed water to form a double bond. Then we carried out the reduction of the double bond to a single bond. So now we've got the framework for butyric acid, the 4-carbon atom molecule. So this is really the end of one cycle. So let me just put here, that's really the end. And now I'm going to give you a little bit of the next cycle just so you can see how this is going to work here. And so what I'm going to do in the next cycle is I'm going to transfer this part of the molecule onto the this enzyme that we had up there, the KS. So I'm simply going to transfer this whole stuff here onto that KS enzyme. And so here we have CH2, CH2, CH3. So this is the start of the next cycle. We end the first cycle here. We're going to start the next cycle. So this looks just like this over here, but it is now 2 carbons longer. So we bring in to the cycle another molecule of a malonyl group. We reload the acyl carrier protein that used to be here. We move that over here, bring another molecule of malonyl onto here. And so this is going to be our malonyl group here. I'll get this right. So another molecule of malonyl has come in here. The same thing happens here. These electrons attack that carbonyl. That goes up there. And we're going to generate again the empty KS. And over here, we're going to have the acyl carrier protein. Now is going to be this molecule here. 2, 3, 4, 5, 6, and we release the CO2 that used to be there. Okay? So now we're beginning that second cycle. So that's the overall pathway. It looks similar in some of its chemistry to the breakdown. The primary differences in terms of the chemistry is for every step we have to use this activated malonyl coenzyme A, and then these internal steps are actually rather similar. So that's the overall process. If we now want to form the palmitic acid, how many cycles do we need to go through? Palmitic acid has 16 carbons. That's 8 pieces of acetyl-CoA. We have to go through 7 cycles to form those 8 pieces in with 7 bonds between them, or 7 attachments between them. Okay? So we have 7 cycles. And when this particular enzyme, generally when it gets to palmitic acid, it says that's enough. It measures the length of it, that says that's what I want to make, kicks it off. Okay? So that's the end of the show right there.
I have a question.
Yeah.
You said for every step you have to use what?
1 molecule of malonyl coenzyme A. We start with 1 molecule of acetyl-CoA, but then for every step we have to add another molecule of malonyl coenzyme A. Okay? Okay, let's do the summary equation now. So here we're going to have, at the very beginning, we started with 1 molecule of the acetyl group, and it starts as acetyl coenzyme A. So there's 1 molecule of acetyl coenzyme A plus 7 molecules of malonyl coenzyme A. We go through 7 cycles. Each cycle requires 2 NADPHs, so that's going to be a total of 14 NADPHs that are required. There's 14 protons involved here, and this is going to make our molecule of palmitate. It's going to give us 14 NADP plus molecules. It's going to give 7 molecules of carbon dioxide. Plus 8 molecules of coenzyme A, 1 from this coenzyme A, 7 from that one, and I think 6 waters. So this is the overall summary equation here. If we go back now and remake the malonyl that we started with, so this is the balanced equation for the reaction proper. But now let's count the malonyl reaction here and the synthesis of the malonyl. If we start from 8 molecules of acetyl coenzyme A, then we need 14 NADPH molecules, and we need 7 molecules of ATP, 1 for each of the malonyls in order to synthesize that. 7 protons, and then that will make us our palmitate C16 molecule, 14 NADP pluses, 8 molecules of free coenzyme A, and 6 waters, and 7 ADP plus 7 phosphate. So starting from the very beginning, using nothing but the acetyl coenzyme A molecule here, we would need 14 NADPHs and 7 ATPs in order to complete the synthesis of the 16-carbon palmitic acid. Okay? So that's going to be the overall balanced equation for this process. And you can make fatty acids of different chain lengths by simply adjusting this. Generally what happens is that this one enzyme usually goes in most organisms to the C16, and then other enzymes will lengthen that further. So that's generally the way in which it will happen. Any questions on the overall process here that we've got?
Where was the malonyl-CoA synthesized in there? Like, where is it regenerated?
It's over here. So if we start with the acetyl coenzyme A, if that's our starting point, then to make the malonyl, we need 1 ATP and 1 CO2. Okay? And so if this balanced equation here is just starting with malonyl, but if we start with acetyl, we have to include the reactions to synthesize those 7 malonyls, and that's where the 7 ATPs come in. Okay?
And there's no release of carbon dioxide on the bottom one just because of it. You didn't look that offering image, it's just.
That's right. We've already taken care of that.
Okay.
Because we've well, by adding 7 of these reactions to one of these, we should get that, I hope. Yeah, that's the idea. We add 7 of those to one of that, and we should get that. Okay? So that's the overall balanced equation. I think I just had a couple of little more things here. Yeah, this is the enzyme. This is truly an amazing enzyme. This is the enzyme that the fatty acid synthetase enzyme, and the structure has been worked out, and this is just kind of a cartoon of that structure. And what you can see here, let me bring this over here so you can see that. And here we have the acyl carrier protein with its flexible thing, okay? Its flexible arm with the sulfhydryl. And this is a dimer. Everything on the left is mirrored by everything on the right, so we only have to look at one side of this. We start by attaching the malonyl coenzyme A to the acyl carrier protein, and then we attach the acetyl group to the KS enzyme, which is right here. That comes over and forms the condensing reaction that you see right there. Then it carries it through these different enzymes that are given different colors here, and they represent each of these steps along the pathway. So it's kind of like an assembly line. Each enzyme carries out a specific step in the reaction, and that acyl carrier protein carries the growing fatty acid through those different enzymes to allow the reaction to occur. So it's really a pretty amazing little enzyme. Okay. Any questions on that then?
What did the CE down here stand for?
That's the condensing enzyme. And that's not, I think that might be an old nomenclature, and what your textbook uses is the KS for that condensing enzyme.
Okay.
I think that's the difference there. I think if you go back, if I've had a few nomenclature differences, well, that just gives the names. It doesn't give the abbreviation here. It might give the abbreviation in that. Okay. Well, I think this is a good place to stop. I would be willing to work 1 or 2 problems on the old exam if anybody wants to watch that for the next couple of minutes. I'll just work one very quickly that's directly related to this, and that is, if you have the fatty acid, this is question 4 on 2011, if you have the fatty acid C12, okay, 12 carbons, how many ATP molecules are produced by the complete metabolism of this molecule? So this is a question which is directly related to what we just talked about in the fatty acid degradation. So let's take a little look at that. So we're starting out with C12, and we want to know how many ATP molecules are produced. And it's going to be a ton of ATP molecules, as you can imagine. But what we're going to have here is going to be, let's just write out the C12 here. Show you what that looks like. So we have CH3, CH2, 10. That's the fatty acid. We first activate that with coenzyme A to make the coenzyme A molecule. Okay, and we also make AMP plus pyrophosphate, and that goes to 2 phosphates. That's where we use that energy of ATP twice. Okay? Now, in the next step, we're going to take the coenzyme A, CH3, CH2, 10, and we're going to undergo how many cycles? This is the key thing. 12 carbons, okay? That means 6 pieces of acetyl coenzyme A are going to produce how many cycles? 5. That's once you got that, you got it made. So that's going to be 5 molecules of FAD plus 5 molecules of NADH plus 5 molecules of coenzyme A plus 5 waters. And this is then going to generate 6 molecules of acetyl coenzyme A plus 5 FADH2s plus 5 NADHs plus 5 protons. And then we just do the same arithmetic that we just did. The 5 times 2.5 is going to be 7.5 ATPs. The 5 NADHs times 2.5 is going to be 12.5 ATPs. So that's going to be this acetyl-CoA. When that enters the citric acid cycle, each one of those makes 10. So this is 60 ATPs, and the total number is going to be equal to 80 ATPs. And if we subtract the equivalent of 2 for the priming reaction, that is going to bring us down to 78 ATPs. Okay? And so that's what the answer is for this particular question. So it's just like we did with the C16. You have to first of all figure out how many cycles you have to go through, and then do the metabolic accounting to get the total number of ATPs that are produced.
Where did you say the 2 at the end?
Well, this first priming reaction here uses 1 ATP, but the equivalent of using it twice, because it's going to AMP and pyrophosphate. So that's why you subtract 2.
Where do you multiply those by? Where'd you multiply at the very bottom? Like, get 12.5, you get 7.5.
Well, each FADH2 in the respiratory chain is 1.5.
Okay.
Each NADH is 2.5.
Okay.
Okay. Okay. Have a good weekend, man. See you next Tuesday.